Hello, this is lecture number 37. This is on the biology of maternal aggression. And this is an area of research in the study of maternal behavior that I literally have devoted my entire career to studying. You have an ebook that I've sent to you that I wrote a few years back, and you've been reading that, and hopefully it's been insightful in terms of your learning about this very interesting aspect of maternal behavior and the biology of this type of behavior. And I've also sent you a videotape that I shot a number of years ago, which I think illustrates very well the intense nature of this behavior, a mother protecting her offspring from an intruder male that was placed into her cage. So again, this is a behavior that has been noticed in many different mammalian species, but up until the time that I started studying it, which now goes back around 40 years ago, again, spent an entire career studying it. Hopefully that book and the discussion that we have about this behavior will help you understand a little bit more about how a behavioral neuroscientist actually goes about doing their work, because it's work that requires a lot of patience. It's work in which you have a lot of failures. It's work that you get to answer some interesting questions along the way, and it's work that's really never complete. There's still so much to be learned about this very interesting behavior, but we will take a look at it. Maternal aggression is the defense of newborn offspring, which obviously serves a very important role. That role being protecting the young and making sure that they develop normally. There's males that potentially would want to invade the nest site of a female and kill those offspring and then mate with the female in order to have their own genes passed on. So this is something that is seen in almost all mammalian species. If you have pets, maybe you've seen it in a pet dog or a pet cat that you've had that has delivered some offspring. So again, it's a very important behavior in terms of ensuring the normal development of offspring. So if you take a look at this behavior, and again, you saw a videotape, you watched a videotape of this behavior, but this is a female mouse that you see right here that's hovering near her offspring. She's built obviously a beautiful nest for them. And those are pups that are probably about somewhere between 6 to 8 days following delivery. And here's a mother nursing her offspring. She's in a nursing posture, as you see there. Here is where a male has been introduced to the cage of this female. This male has been marked with a magic marker so that we can distinguish it easily. And the female rapidly lunges towards the male and then begins this incredibly intense attack behavior. And again, that videotape that you saw should help you to appreciate, how intense this behavior really is. So again, this is the behavior that we are studying, and it's one in which, we've been able to answer, I think, a number of important questions. So there are key questions here. These are the questions that we're going to be addressing. We want to know, what role the young actually play in this type of aggressive behavior seen by females shortly after they deliver their offspring and continuing on while they care for their young and up until the time that they're weaned and go off on their own. We want to know the physiological events during pregnancy that may be responsible for this type of aggression. We also want to know what physiological events following the delivery of young may be responsible for this aggression. And we want to know also, are there some early developmental factors that might be involved in this type of aggressive behavior? By that I mean maybe there's something, as in the case of sexual behavior, for example, where we know that the brain is differentiating in important ways. Maybe there's something that's occurring in the female that's really important during that early fetal period of time that is critical for that female to exhibit normal levels of maternal aggression later on in life. And I think that certainly we want to know about genetic factors that might be involved, that might influence this kind of aggression. So these are just a few of the questions that we've explored in their through the years, and they are questions that we'll try to answer over the course of this lecture. So one of the first things that we did was to explore changes that occurred in aggression that were associated with the reproductive state. And what we did was we simply took some female mice and mated them, and they became pregnant, and we assessed what their aggressive behavior would look like when we took a male and placed them into their cage over the course of their pregnancy and then after they delivered their offspring. So what you see on the top portion of that slide is what happens in terms of their aggression. And one of the things that we saw was that beginning around mid-pregnancy, and again, the timeline here is this is days of pregnancy that you see here. We call it gestation. And this is after the mother delivers her offspring. This is lactation. Up until 22 days postpartum. So what we're doing is we're simply looking at what happens when a strange male mouse is placed in their cage, and you can see that early on in pregnancy very little happens, but as the female gets into about mid-pregnancy, they begin to show this threat kind of behavior where they will lunge towards the male, but it actually stops short of any kind of physical contact. Then, after the mother delivers her offspring, there's about a 24-hour to 48-hour period of time where they don't exhibit any aggression. If you were to take a male and place it into her cage, she would, in fact, not exhibit any aggressive behavior towards that male. In fact, what she would probably do is mate with that male. It's not uncommon in the wild to see animals that are taking care of offspring, but also carrying, that is, they are pregnant with another litter. There's this period of time, it's called postpartum estrus, where the female becomes very receptive to a male. But then you can see, beginning at about 48 hours, 2 days after she delivers, the behavior that she exhibits towards a strange male becomes intensely aggressive. And peaks usually around day 12, between day 12 and day 14, then progressively starts to go down as the young get older. So this is something that, this is one of the first things that we did to explore this behavior. And then what we also did was we started to explore what would happen to what happens in various hormone profiles during pregnancy and lactation. So let's look at these three major hormones that you see here. Here's progesterone that you see here, this line. And you can see progesterone is really going up very substantially around mid-pregnancy. Then drops very, very low shortly before the mother delivers, and then remains low until the latter part of the period of time when she's caring for her young. That's progesterone. Estrogen, on the other hand, you see it's very low, but during most of pregnancy, but then escalates, begins to go up right prior to the time that the female delivers, and then stays high for the first 24 to 48 hours, and that's a period of time that is called this postpartum estrus. And estradiol, no doubt, is very important for that postpartum estrus that you see there. And then we take a look at the hormone prolactin, and prolactin really shoots up right before the female delivers her offspring, and then goes up even higher right after the female delivers. She's starting to receive suckling stimulation from her offspring, which is going to cause the release of even more prolactin. Prolactin stays very high till about mid-lactation, and then begins to go down. So note how similar this is with this that you see right here. So prolactin automatically became a hormone that we were interested in exploring. Some of the first questions were, again, very simple ones. What's the role that the young play? And you can do a very simple experiment, one that we did, in which we took the pups away for a 5-hour period of time, or for just a 1-hour period of time, or we took the pups away from the mother and then tested the mother immediately after we took the pups away. And these are all females that were had been with their offspring for about 4 to 8 days. So they had been with them for a period of time. And again, what you see here is that if you take the pups away for a 5-hour period of time, this aggressive behavior goes to near zero. Taking them away for 1 hour, not a big change. And again, simultaneously taking the pups away and testing for aggression, no change at all. 100% of the animals are aggressive. So again, this was a very important study, very revealing study. I mean, it shows clearly that the presence of the young during this postpartum period are really important for the behavior. If the young are not around, even for a shorter period of time as 5 hours, the aggressive behavior of the female goes way, way down. So again, this is here's our mother here. That's she's obviously in close proximity with her offspring, nursing them periodically. As long as that is maintained, then you continue to see this very intense kind of aggressive behavior. But then we asked another question: what would happen if we took pups away for 5 hours and then we replaced them, put them back in the nest for a period of, let's say, 10 minutes, and then tested the female, or for a period of 5 minutes and then tested the female, or if we simultaneously placed the pups in, again, after they had been away for 5 hours, and tested the female for aggressive behavior towards a male. So this shows the duration of pup replacement. These are all in aggressive females that have had their young taken away for 5 hours. And what you see is that as little as 10 minutes of reinstatement, that is 10-minute period of time, when the female is back with her offspring, you get this, 100% of the animals exhibiting aggressive behavior. After 5 minutes, it's not quite as high, but just putting them back in and simultaneously testing the female for aggression, no aggression. So the animals have to be with the young for at least 5 to 10 minutes. 5 to 10 minutes of pup replacement in order to reinstate that aggressive behavior after they've been away for 5 hours. So there must be something very immediate that is occurring physiologically in order for that behavior to be restored like it is. So again, this is a very interesting finding, and it's one that we now wanted to pursue in a little bit different way. And I think that that's portrayed in this slide that you see here. What we did was we took females and put them, aggressive females, ones that we knew that were exhibiting this maternal defense or postpartum aggression at very high levels. And what we did was we allowed those females, some of them, to stay in direct contact with their offspring. That is, we didn't remove the offspring. And in another condition, what we did was we used a partition in the cage that the female was in with her offspring. And we took the offspring and we put them on the other side of a double wire mesh partition, so the mother could see the offspring. She could hear them, she could smell them, but she couldn't have any direct contact with them. And in yet a third condition, what we did was we took the pups out of the cage completely, so that there was no contact. And indeed, what we found was that females that were in direct contact with their young, once we placed a male into the cage, they showed very vigorous aggression. Same was true in the case of females that no longer were in direct contact but could see, smell, and hear their offspring. And again, our control condition here, where the pups are no longer there at all, they're not behind the partition, they're not in the cage. Again, very little aggression. So this indicates that there is some kind of a sensory cue coming from the offspring that's important for maintaining the aggressive behavior of the female. So this was an interesting line of experimentation that helped us to understand this behavior. So let's go back to this figure where we take a look at what is happening here in terms of this timeline where you're getting this threat behavior that we see during pregnancy, then this period of time when the female's not aggressive at all, and then you get this very intense attacking behavior which begins about 48 hours after. We began exploring the possible contribution that progesterone might be playing to this behavior. Behavior that during pregnancy, that this very interesting threat behavior, which really actually falls short of actual attack behavior. And what we did is we started treating virgin female mice. These are mice that had not been through pregnancy at all. We started treating them with progesterone. And one of the things that we found was that when we started treating these virgin females with progesterone, that after a period of time, they would show this kind of threat behavior. Here you can see what happens over an 8-day test period where the animals are being tested every day. Animals that are treated with this progesterone, you're seeing this threat behavior increasing, unlike what you see in control animals that have not been receiving progesterone, but instead have been receiving a control injection of sesame oil. So this is very interesting. It suggests that progesterone may be playing a very important role here in terms of activating this behavior during pregnancy. Again, go back to this very interesting figure here. Again, take a look at what's happening with progesterone. Indeed, that escalation in progesterone may be what is responsible for this kind of threat behavior that we see here. We then began to explore, what is happening during this postpartum period of time where you're seeing this really very, very intense aggressive behavior. And one of the things that we learned was that when a mother receives nursing stimulation, suckling stimulation from her offspring, there are some really important events that occur. When a suckling stimulation is applied to the nipple area of the female and any mammalian female, it activates these spinothalamic tracts that you see here. You're getting neurotransmitter changes that are occurring in the supraoptic and paraventricular nuclei, the hypothalamus, which in turn is resulting in these hormones being secreted by the pituitary gland, lactogenic hormone, which we also call prolactin, responsible for milk, for the development of milk in the mammary glands, milk secretion. And then oxytocin, which is responsible for milk ejection, in the mammary gland. So again, we have some really interesting neurotransmitter changes that are occurring here, and things like serotonin, for example. as well as in changes that are occurring with prolactin and with oxytocin. So we started focusing in on those changes as being important ones. And one of the things that we started to do was to do some simple experiments where we took females, and before they delivered their offspring, we surgically removed their nipples so that they couldn't receive suckling stimulation any longer. And one of the things that we found was that when we did this, these females failed to deliver, failed to exhibit aggressive behavior after they delivered their offspring. So we felt then that suckling was absolutely crucial for turning on this behavior. So again, here's a female mouse that's suckling, receiving suckling stimulation from her offspring. She's suckling from they're suckling from the abdominal area that you see right here. And this female would exhibit normal aggressive behavior, but if she didn't have her nipples, if they had been surgically removed, hence she couldn't receive suckling stimulation from her offspring, she would fail to exhibit aggressive behavior. So this was a paper that we were fortunate enough to be able to publish in one of the most distinguished scientific journals called Nature. And there I am with my co-author right there. This was the paper that we published in Nature. And again, that was a paper that's been highly cited by others working in this area and proved to be, a very important finding. We found, for example, that if we were to take females, virgin females who could not receive suckling stimulation because they hadn't gone through pregnancy. One of the things that happens during pregnancy is that the nipples grow as a consequence of the secretion of estrogen and progesterone. We found that if we took virgin females and started to treat them with estrogen and progesterone in order to grow their nipples, to allow them to be able to receive suckling stimulation, that those females, once they receive that suckling stimulation, would exhibit normal levels of aggressive behavior. So that was a very important finding in this area. So the virgin female mice could be induced to show aggressive behavior when they were given hormones that cause the nipples to grow. Again, give them pups, they hover over their pups, they start to receive suckling stimulation, and as a consequence of that, they can now exhibit normal aggressive behavior. So this was an important finding in this area. And then we started to ask, well, what's that suckling doing? And one of the things that we wanted to focus in on was, of course, prolactin, which many have considered to be the maternal hormone for a very long period of time. And we started to do some experiments where, for example, we would take females that had delivered their offspring, and we would start to treat them with this drug called bromocriptine, which suppresses prolactin release. And essentially what we found is that there was no effect at all of this manipulation, that they exhibited normal aggressive behavior even though they were not secreting prolactin any longer. We did experiments where we actually removed the pituitary gland of females shortly after they delivered their offspring. What we found was that these females continued to show completely normal levels of aggression. So that suggested that really prolactin and probably oxytocin are really not involved in this behavior at all. What you can see here is here's the pituitary gland, and to surgically remove the pituitary gland, this is in a human, but in a mouse, what you have to do is you have to go through the ear canals in order to do it, and it's something that is it takes a long time to learn how to do this and learn how to do it accurately, and this occupied a good 2 or 3 years of my research career in order to perfect this, to be able to do it in the case of mice. So doesn't look like prolactin's involved, doesn't look like oxytocin is involved, but then we started to explore the contribution that neurotransmitters might be playing. And a good candidate for this was this neurotransmitter serotonin, which we know dramatically changes in the brain, been implicated in mood changes, both in humans as well as in lower animals, implicated in aggression. And what we did is we started treating female mice with drugs that would inhibit the release of serotonin. And this is one such study where animals were treated with a drug that's called PCPA, which suppresses the secretion of serotonin. Another drug we used was methosergide, and again, under both situations, and in quite significant reductions in the ability of females to show this kind of aggressive behavior. Here you see what happens with this dosage of PCPA, and here's what you get with this particular dosage of methosergide. So this really suggested to us that there's an important contribution to the turning on of this behavior, at least in the mouse, by suckling-induced activation of the serotonin system. Other researchers, namely this one that you see here, Stephen Gammy, has implicated another hormone called CRF, corticotropin releasing factor. And he believes that that ordinarily suppresses maternal aggression in postpartum mice, because one thing that happens with a mouse is after they deliver offspring, CRF goes way, way down, okay? And indeed, he believes that that's a trigger for aggressive behavior. So if you start giving a female mouse, a postpartum female mouse, even small doses of CRF, it'll cause a suppression of their aggression. And indeed, this is what he found here. With as little as about 0.2 of a microgram of CRF, you're getting reductions in aggressive behavior. Again, here, a reduction in, this is the number of attacks, this is the time spent exhibiting aggressive behavior. He's also implicated another neurotransmitter, nitric oxide, which is a gaseous neurotransmitter. By working with mice that are deficient with that particular neurotransmitter, he finds that these nitric oxide deficient mice don't really exhibit very much maternal aggression at all. And that's reflected in this figure that you see here. In the nitric oxide deficient animals, they're showing reductions in terms of their aggressive behavior. So again, the picture that emerges here is that there seems to be a lot of different biological factors that are involved. We implicated serotonin, he's implicated CRF, as well as nitric oxide. Gammy's also done some wonderful research, taking a look at brain neural activity during aggression. And one of the things that he does is he measures something that's called CFOS. CFOS is a way of indirectly measuring neural activity in the brain. And what he finds is that during aggression, here are the areas that are activated: the olfactory bulb, the septum, the amygdala, and the preoptic area. That's in, of course, the old brain. The periaqueductal gray area, the PVN, paraventricular nucleus of the hypothalamus, the peripeduncular nucleus, the ventromedial hypothalamus. Now, these are all areas of the brain that have been implicated in other forms of aggression. So this is interesting research. It certainly doesn't show, a cause and effect relationship. This is activation that is occurring during an aggressive encounter, but it's very interesting that he has been able to document that this high C-phos brain activity during aggression in these neural areas that we know to be implicated in other forms of aggressive behavior. This piece of research, which just came out actually just a few years ago by Liz Unger, she's been taking a look at both female aggression, maternal aggression, and male aggression, we call it intermale aggression. And she finds some interesting relationships between this, which is called aromatase, which is an enzyme that is needed to convert testosterone into estrogen in the brain. And it seems to do so in neurons in the amygdala. And the amygdala, of course, has been implicated in maternal behavior. It's been implicated in maternal aggression. And here's what she finds. When you take a look at this aromatase, an aromatase containing neurons in the amygdala, this conversion that takes place from testosterone to aromatase is very important for male behaviors like intermale aggression. What she also finds is that in the female, the ovaries also produce a small amount of testosterone that gets aromatized, that is converted to estrogen. And she finds that this is involved in maternal aggression. So this is fascinating new information about the role of testosterone being converted to estrogen that may be important for various aspects of female aggression. Interestingly, it doesn't seem to be involved in any other aspect of female behavior, like female sexual behavior. So this is interesting information, and certainly is giving us more of an idea of the biological underpinnings for this particular behavior. So we had been working with mice that are what we call outbred mice. You see here, these are Rockland Swiss albino mice. And you see enormous variation in their aggressive behavior. There are a small percentage of females that never show aggression. There are some that exhibit just, maybe over 100 attacks in a short like 3 to 5 minute tests. There are some that just show a moderate amount of aggression. Tremendous variation in their aggressive behavior. Maybe it's related to genes. Again, when we take a look at Rockland Swiss mice, they are randomly bred animals. That's why you're getting a lot of variation in aggressive behavior. Inbreeding is really limited to about 1%. So the animals are not identical to each other, and that is why you're getting this tremendous variation. Well, when you begin to take a look at inbred animals, you begin to see, a different, little bit different and interesting pattern that emerges. And we started exploring DBA2J mice and C57 black 6J mice. Again, they are dramatically different in terms of their genetic backgrounds and very different in terms of their biochemistry. These animals, these mice, are by far the most researched mice in the world today. We know a great deal about them in terms of their behavior and in terms of their physiology. So we started using them in our work as well. So it's interesting to take a look at the goals of psychology and behavioral neuroscience in the context of this. So we're interested in predicting and controlling behavior. We're interested in understanding individual differences in behavior. Why is it that individual humans and animals can respond in such different ways to the same social situation? So indeed, that is something that we can do by focusing our attention upon working with inbred animals. We can begin to learn the degree to which genes may be playing a role in variation that we see. So again, this comes down to how much of our behavior is nature, meaning genes and hereditary factors in terms of not only physical appearance, but behavioral appearance, and nurture, these environmental variables, childhood experiences, and so on. We want to understand about each of these, nature as well as nurture. So, postpartum aggression, when we begin to take a look at it in these DBA and C57 black mice, one of the things that we find is that when we look at DBA animals, that's this brown mouse, they show very, very high levels of aggression. When you look at the C57 black mouse, as you see here, they exhibit very low levels of aggression. So when we just started contrasting and comparing this behavior in these two very well-known genetically diverse inbred animals, we saw, dramatic differences between the two of them. When we took a look at pregnancy-induced aggression, what we found was that the DBA female, very, very aggressive. The C57 female, somewhat aggressive, but not anywhere near as much as what we saw in the case of the DBA animal. So again, dramatic differences between the two. We began to explore these two different inbred strains in terms of what we knew, what we could learn about progesterone. And this graph that you see here shows DBA and C57 animals in terms of their aggressive behavior and in terms of their progesterone values. And what we found was that there was no real dramatic difference between them in terms of this hormone progesterone over the course of pregnancy. But of course, differences that you see here in terms of their exhibition of this threat behavior during pregnancy. So we conducted other studies in which we began to explore what would happen when we started exposing some virgin females from each one of these strains to different dosages of the hormone progesterone by implanting these very small celastic capsules containing this hormone underneath their skin. And what we found was that DBA animals were much more responsive to receiving that progesterone in terms of them exhibiting aggression in comparison to that of C57 animals. So again, that suggests that perhaps there's something about the way in which the brain is responding to that hormone that is different in C57 and DBA animals. Maybe DBA animals simply have more progesterone receptors in the brain. Another important way of exploring this individual variation is to take a look at prenatal stress. Prenatal stress is an important source of environmental variation. Prenatal stress can do a number of different things to result in changes in terms of brain development. Prenatal stress can change the fetal hormone production. It can change patterns of brain development, and those changes can result in changes in behavior later on in life. So you see a fetus developing here. Here's the placenta, and the female, the fetus, of course, is deriving not only nutrition from this, but is also being exposed to various hormones during this early developmental period. So we began to explore this then as an important source of variation. And one of the things that we found was that when we expose animals to prenatal stress, as you see here, a pregnant female restrained in this situation and put under bright lights, 3 times a day for 45 minutes per day. Do this during the last 11 days of her gestation. One of the things that we found was that pregnancy, once these animals delivered their offspring, and we took a look at the females, allowed the female offspring to grow up and then mated them, and took a look at their aggressive behavior during pregnancy and during the postpartum period. We found that females that had been stressed prenatally exhibited reductions in terms of pregnancy-induced aggression, but elevations in terms of their postpartum aggressive behavior. So indeed, this was very interesting, that this environmental stress during pregnancy, could indeed modify their behavior later on in life. What the source of that is something that we didn't really know. We thought that maybe it was related to the amount of testosterone, perhaps, that they were being exposed to. But it could have been just about anything. We then started to explore, as another very important source of variation, this prior intrauterine location. During pregnancy, while a female is carrying fetuses, a female mouse can carry anywhere between, probably about 6 to 20 fetuses. And these are the 2 different uterine horns that we see here. These rodents are what we call politicus, that is, they carry many different offspring over the course of a pregnancy. And you can classify these female fetuses based upon their proximity to males. So, for example, this female that you see here is classified as a 0M female because she's not surrounded by any males. This is a 1M female because she has, this female has a male here and a female here. This is a 2M female because there's a male on either side of her. So we can classify them based upon their proximity to a male. What we did was we cesarean delivered these female fetuses on the 18th day of pregnancy. We noted their location in terms of who they were proximate to. Were they proximate to either males or females? And then we started to explore them in terms of their behavior. And indeed, we found some very, very interesting things. If you take a look at this very important marker called anogenital distance, in humans, here's how we measure it. Here's a male that we see here. We measure the distance from the anus to the genital area. So this is what we see in a male. What we see in a female is a much shorter anogenital distance. And this is due to the action of testosterone. Testosterone is present in early life, as it is in the case of a XY condition, a male. It works on this area right here, and this area, this distance is much longer than it is here. So this is a way, really, and this is a female that you see here. This is a way of really noting the effect of using as a biomarker. This is a measure of masculinization. And if you take a look at it in terms of rodents, for example, here you see this anogenital distance right here in the case of a male, and here we see it in the case of the female. This is much shorter than this. And again, this is a measure, really, of what is happening during early life, what is happening during fetal life, where males ordinarily are exposed to testosterone and females are not. So here's something that we did. We took a look at intrauterine location and this aggressive behavior. And what we also did was we measured this anogenital distance at the time of birth. And we also measured levels of testosterone in circulation. And what we noted was that females that were not surrounded by any males, and then they were allowed to grow up into adult life, and then we mated them, and then took a look at their aggression. They exhibited very low pregnancy-induced aggression and very low postpartum aggression. What we found in 0M animals is that their anogenital distance was also quite short. When we took a look at 2M females, these are females now that are surrounded by 2 males, their aggression was very high and their fetal testosterone was high, and they had long anogenital distance. It was almost as if these 2M females had been at least partially masculinized by virtue of their exposure to those 2 males. Males that were surrounding them, that testosterone was probably getting into the circulation from these males into the female, and indeed it was probably producing some degree of masculinization. Now, lastly, we took a look at what would happen if you were able to raise a female in a situation where it was not exposed to any other fetuses. And this is a situation that we call the Singleton female. And the way in which we did this was to not allow other fetuses to develop next to these females in utero. And we did this by surgically removing all other fetuses and just allowing one female to develop in utero. And we compared it to females that had no other surrounding females, no other surrounding males. So our 0M condition, this is a female now surrounded by 2 females. And what we found was that the 0M females had some fetal testosterone. They had short anogenital distance and very low aggressive behavior. But when we took a look at our Singleton condition, what we found was that there was no fetal testosterone, very short anogenital distance, and no aggression. This graph that you see here is a reflection of this. Our Singleton animals showing very, very low levels of aggression. No aggression really in comparison to 0M animals. So again, this really does suggest, in combination with some other work that we did in which we actually injected very low levels of estrogen to pregnant females while they were carrying female fetuses, that this indicates that testosterone may be a very important factor, that it's at least partially masculinizing the brain of a female during early life. So our conclusions, certainly the presence of young, very crucial factor in terms of the initiation as well as the maintenance of postpartum aggression. We also noted that progesterone is important for stimulating pregnancy-induced aggression. We also noticed that suckling stimulation induces changes in serotonin, probably other hormones and neurotransmitters as well, and this is crucial for the development of this behavior. And we also noted that prenatal testosterone appears to be elevating aggressive behavior, and genetic factors, things like prior intrauterine location as well, seem also to be very much involved in this behavior. So why is these studies important? Well, from a basic research perspective, they are important. It helps us to understand the biology of this behavior, which is obviously very crucial for defending one's offspring and making sure that they are out of harm's way, and make sure that they are going to develop to a point where they ultimately are going to be able to become reproductively active later on in life. It's also important for us in terms of trying to understand the nature of aggression, where at one point we thought that this was not something that females ordinarily do, that they don't have a brain that's wired for aggressive behavior, but we certainly know differently now that the females, depending upon their reproductive condition, are every bit as aggressive, if not more aggressive, than that of a male. This kind of work also, I think, is helpful in understanding mood changes in women. The work that we talked about earlier when we were looking at maternal behavior certainly is work that has helped us to understand mood disorders like postpartum depression, for example, and postpartum blues syndrome. So I think for all these reasons, this work is important. So that really wraps up our study of maternal behavior, and our next class will begin to take a look at laterality and the fact that the two hemispheres of the brain are serving different functions.